Have you ever wondered what makes you, you? What gives you your unique traits and characteristics? It's a fascinating question, isn't it? The answer lies in the realm of genetics, a science that explores the very essence of life. Genetics is the study of heredity and the variation of inherited characteristics. It's the reason you have your mom's eyes or your dad's laugh, why you love the taste of cilantro or hate the sight of spiders. But it's not just about physical traits. Genetics also plays a role in shaping our behaviors, our tendencies, our quirks. At the heart of it all is DNA, a molecule composed of two chains that coil around each other to form a double helix. This is the blueprint of life, containing genes that dictate everything from your hair color to your susceptibility to certain diseases. Well, it's all written in a complex code within us, a code called DNA. The journey to unraveling this code began in the 19th century. In the heart of Europe, an Augustinian monk named Gregor Mendel was meticulously studying pea plants. Mendel, often referred to as the father of genetics, was the first to notice patterns in the way traits are passed down from generation to generation. He conducted thousands of crossbreeding experiments, tracking 7 different traits such as plant height and seed color. He discovered that certain traits can disappear in one generation, only to reappear in the next. This was the first glimpse into the world of heredity. But it wasn't until the mid-20th century that the physical basis of heredity was discovered. Enter James Watson and Francis Crick. These two scientists, working out of a small laboratory in Cambridge, England, were determined to crack the code of life. They were searching for the structure of deoxyribonucleic acid, or as it's more commonly known, DNA. DNA is a large molecule that carries the genetic instructions used in growth, development, functioning and reproduction of all known living organisms. But its structure was a mystery. Watson and Crick, building on the work of Rosalind Franklin, Maurice Wilkins and others, proposed that DNA is a double helix, a twisted ladder shape. They suggested that the steps of the ladder are made of pairs of 4 different chemicals called nucleotides which always pair in the same way, adenine with thymine and cytosine with guanine. This structure explained how DNA can be copied when cells divide, and how the instructions it carries can be used to build proteins. It was a breakthrough that revolutionized biology and medicine. The discovery of the DNA double helix was announced in April of 1953, and Watson, Crick and Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962. And thus, the structure of the DNA helix was revealed, opening doors to a whole new world of genetic exploration. Understanding the structure of DNA was just the beginning. Now let's dive deeper into the real magic behind genetics, the process of DNA replication and the role of the 4 nucleotides, adenine, thymine, cytosine, and guanine. Imagine DNA replication as a process where a DNA molecule creates an identical copy of itself. Picture 2 strands of DNA, intertwined in the famous double helix. They separate, making room for new nucleotides to attach themselves and form new strands. This results in 2 DNA molecules, each a perfect replica of the original. But what about these nucleotides we just mentioned? Well, they're the building blocks of DNA. Each nucleotide has one of 4 bases: adenine, thymine, cytosine, and guanine. These might seem like random names, but in the world of genetics, they're the equivalent of the alphabet. Adenine always pairs with thymine and cytosine always pairs with guanine. They're like best friends, always sticking together. This pairing is crucial. It's the foundation of how DNA passes on genetic information from one generation to the next. Now this is where it gets interesting. The order in which these nucleotides are arranged forms a unique genetic code. Think of it as a string of letters forming words, sentences, and ultimately, the chapters of a book. This book is then read by our cells to build and maintain our bodies. The sequence of these 4 nucleotides is what makes us who we are. It determines our physical traits like the color of our eyes, the texture of our hair, and even our susceptibility to certain diseases. Every human being has a unique sequence, a different story written in their DNA. So the next time you look in the mirror, remember that you're looking at a unique genetic story, written in a language of 4 letters that took billions of years to compose. Just like a unique string of letters forming sentences in a book, these sequences spell out the story of our genetics. Today, we're not just reading this genetic book, we're learning how to edit it. Welcome to the thrilling age of genetic engineering, a revolutionary field that's turning science fiction into reality. Genetic engineering is essentially the manipulation of an organism's DNA to achieve desired traits. Imagine being able to eradicate genetic diseases, increase crop yields, or even create bioluminescent plants. Sounds like something from a sci-fi movie, right? But with genetic engineering, these possibilities are inching closer to reality every day. Let's take a moment to understand the mechanics of this process. It starts with the identification of the gene of interest, the piece of DNA that codes for the trait we want to alter. This gene is then inserted or edited in the DNA of the organism using various techniques. And among these techniques, there's one that has been causing quite a buzz in recent years. CRISPR, or clustered regularly interspaced short palindromic repeats, is a technique that allows scientists to edit genes with unprecedented precision and speed. It's like a pair of molecular scissors, capable of cutting DNA at specific locations to add, remove, or alter a sequence. This has opened up a realm of possibilities in medicine, agriculture, and beyond. In medicine, for instance, genetic engineering could be used to treat or even cure genetic diseases. Imagine a world where diseases like cystic fibrosis or muscular dystrophy are things of the past. In agriculture, it could be used to create drought-resistant crops or crops that require fewer pesticides, addressing food shortage issues in parts of the world. But like any powerful tool, genetic engineering comes with its fair share of debates and ethical concerns. The potential for misuse and unforeseen consequences are real. But with responsible use, the benefits could be immense. It's like having a genetic typewriter enabling us to potentially rewrite parts of our genetic story. The age of genetic engineering is upon us, and it's set to redefine the boundaries of what we thought was possible. So, where do we go from here? What does the future hold for genetics? These are questions that, as we delve deeper into the world of genetics, become increasingly pertinent. The future of genetic engineering holds untold potential, but it also brings with it a host of ethical implications that we must grapple with. Firstly, let's talk about personalized medicine. This is the idea that, by understanding an individual's genetic makeup, we can tailor treatments that are specifically suited to them. No more one-size-fits-all medication. Instead, imagine a world where your doctor could prescribe a treatment that is uniquely designed for your genetic profile. This could greatly improve the effectiveness of treatments and reduce the risk of adverse side effects. Next, we have gene therapy. This is a technique that could potentially allow us to treat or even cure genetic diseases. By altering the genes directly, we could correct genetic disorders at their source. The potential here is enormous. Diseases that have plagued humanity for generations could become a thing of the past. However, with such power comes great responsibility. And this brings us to one of the most controversial aspects of genetic engineering: designer babies. The idea of being able to choose the genetic traits of our offspring is a contentious one. While it could potentially eliminate hereditary diseases, it also opens the door to a world where physical, intellectual, or even personality traits could be selected in advance. This raises a multitude of ethical questions about the value of human diversity, the definition of normal, and what it means to be human. The power of genetics holds promise and peril. As we stand on the brink of this new era, we must decide how to wield this power responsibly. The future is a vast frontier, and genetics is our latest vehicle of exploration. But as we push forward, we must also look inward and ensure that our moral compass guides us through the uncharted territory ahead. So, we've taken a whirlwind tour of the world of genetics. We journeyed back to the pivotal moment of the discovery of DNA, the blueprint of life. A molecule so intricate and unique, it holds the key to our individuality, and yet it's universally shared across all living beings. We then delved into the complex world of the genetic code, a language written in the 4 letters of DNA, which instructs the very essence of life. It's like the universe's most complex software, running on the hardware of our bodies. We explored the age of genetic engineering, an era where we've begun to tweak this code to our advantage. It's a testament to human ingenuity, reshaping life as we know it. Finally, we peered into the future of genetics, a realm of infinite possibilities and challenges. As we continue to unravel the mysteries of DNA, we come closer to understanding not just what makes us, but also the immense power we hold in our genes.